start the class?
Yes, sir.
Listen, every day I am getting few challenges like time management. Still here. Shall we start?
Yes, sir. Yes, sir.
Okay. See, first 10 minutes, class is only for 50 minutes. Out of 50 minutes, 10 minutes are wasted for your stability. You know that fact? So I left with 40 minutes. In 40 minutes, I will take around 10 minutes for revision, mandatory revision. If not, you are not connecting back with the previous lecture. Then to discuss the new topic, I am getting hardly 20 to 25 minutes. In 20 minutes, I need to finish an entire new topic. Then there will be these attendance issues. You need to be cautious. Time is very precious. Okay. So without much delay, I will start the lecture. Everyone pay attention. Abhishek, Shubhir, Anurag, Karabo, don't use mobiles. Sahil, pay attention everyone. So today we'll discuss about AML CML. I need to complete this topic, okay? And Friday I think you are free, right? Up to 11:10. Shall we have?
Yes, sir. Yes, sir.
All right. Okay. We'll see. Fine. Leave it.
We won't see, sir.
All right. Enough. Okay. So see this image, class. When you perform a peripheral blood smear, when you perform a PBS, a peripheral blood smear, and if you see this type of picture, if you see these cells in the microscope, this is a very, very, very dangerous condition. You know what's happening in this? This is a dysplastic cells. What is the meaning of dysplasia? Irregular shapes, irregular sizes of cells, which is a part of cellular adaptation leading to a cancer. Do you remember?
Yes, sir.
Here you see some neutrophils or some white blood cells are small and some are large. Okay, there is no, uniformity among the cells. They are looking small, they are looking large. Okay, this is a dysplastic anemia. If you see this smear, there is a chance that the patient might end his life because this is a cancerous life. Okay. So cancers need to be addressed. That is one thing. Whatever you saw is a myeloproliferative neoplasm. That is a blood cancer, right? A bone marrow cancer. And I want to tell you a very short story. Okay? She is Miss Mary Claire King. Her name is Miss Mary Claire King, Dr. Mary Claire King, sorry. So when she was 15 years old, she lost her best friend. Her best friend has died because of some cancer. She was very sad and she started understanding about cancer. She studied medicine and she is trying to address what is this cancer is all about. Okay. So this is Mary's journey. So she studied medicine and she started studying cancers particularly. And there is one problem with cancers. Keep in mind, cancer starts with genetic level, right? First there are some mutant genes. Mutant genes will cause some cell abnormalities. Cell abnormalities leads to cancers. And each and every cancer has a specific mutations in the genes. Each and every cancer has a specific mutations. And she is started observing the breast cancer individuals. Okay, the women are highly prone to breast and cervical cancer. Even if you say the normal or commonly affecting cancers, there are cervical cancers all around the world. Okay, especially to women. So she started understanding the patients. She is collecting the blood samples. She is studying the genes and their mutations. Then I want to tell what she did. It took almost 20 years for her to understand various types of mutations in the patients. She is collecting cancer patient blood and she started studying the mutations. During her time, again challenges to get shine in a science is very tough. This is her quotation. She thought that working in science is like assisting a male persons. Okay, So they are not appreciating her work, they are not giving her chances. She just became an assistant even though she is a doctor. So she didn't like this clerical job and she left her job and she kept her own research institute. She kept her own lab and started studying cancers. And you know what happened to this Mary Claire? What she did? So Mary Claire has been awarded Nobel Prize, the best prize a scientist can get as a global recognition. And what she did is something remarkable. She was honored by President of America. That means she did something. And you know what she did? She has pinpointed one particular mutation. That mutation is called as the gene. That gene is called as BRCA. This is BRCA gene, breast cancer associated marker. She understood this gene, mutations in this gene lead to cancers among women. Because of her discovery, because of BRCA gene discovery, some billions, 700 million women, almost 1 billion women has been survived from death of cancer. They has been diagnosed, they has been detected in their early forms even before getting cancer to their body. They might get cancer after 5 years. She recognized it because she did a blood test, and that is BRCA gene, BRCA gene, and she has saved 700 million women. You just imagine how the contributions of her. Okay, she is one of the best lady I have ever seen actually. Okay, she is a Miss Mary Claire, and these are the two genes she has discovered. The two genes are BRCA1 and BRCA2. BRCA1 is 65% rate and BRCA2 is 55% among women. And interestingly, BRCA is also present in, it's a breast cancer marker. This is also has been detected in males. The reason how a breast cancer has led to a male anatomy I will discuss later. Genetically speaking, all males are females. Then there are some suppressions of genes leading to a male anatomy. Okay, all males are females when they are embryos. Embryo is a neutral mother. Okay, actually embryo is always a woman. A woman first. There is this societal dominance that male first dominance, but actually genetically or biologically, all living organisms on planet are mothers or their women. So you need to appreciate that we are sharing all women genes and they will sometimes express and that will cause these things. And also, BRCA gene not only associated with breast cancer, this marker is also associated with pancreas cancer, ovarian cancer, and prostate cancer in males. So, BRCA played an important role in understanding the markers, the significance of markers such as genetic marker in this case in order to identify cancers. This is clear, right? Then, you just see, people has recognized her contribution. See the awards she received. How many awards she got? See, all these awards has came to only single woman. And you can understand, how many honorary degrees she got. Okay, these are the contributions of Miss Mary Claire King, Dr. Mary Claire King. Okay, one of the best geneticists we never seen. Okay, maybe sometimes God will send some angels like this for the. And she is still alive. Most of the scientists I know are passed away, but she is still alive. And she recently has took 2 weeks ago.
If it happens to save his life also, that's okay too.
From 1971.
I'm a geneticist at the University of Washington.
The thing that I want to draw here is we are studying the same subject, and I was expecting similar curiosity among you.
In critical genes, the genes
Alright.
carry mutations that lead to very high risks of breast cancer and ovarian.
Alright, so this is all about Dr. Mary Claire. And coming back to our topic, I think because of PPT we are distracted from actual topic, students. Okay, let us keep PPT aside. Let me take any empty slide. There is no empty slide. Alright. Alright, anyway, stop staring at PPTs and pay attention to the board. So, a quick revision. So far, we discussed about aplastic anemias. Everyone write these things, okay? We discussed about aplastic anemias. Then we discussed about leukemias. And we already know the types of leukemias. Then we discussed about the stem cells and stem cells' role in getting cancers. Okay, stem cells' role in getting cancers. We have seen that. That is also important thing. Then we discussed about cellular adaptations. So you already know all those cellular adaptations: atrophy, hypertrophy, hyperplasia, dysplasia, metaplasia, etc. Then we discussed about the diagnosis, basic diagnosis of cancer. Some of the basic techniques we have are, can you tell me? So diagnosis in case of blood, that starts with CBC. Then we saw anemic symptoms in CBC. Then peripheral blood smear. Then biopsy is the ideal candidate. Then after biopsy we have markers, molecular markers we have. Okay, like our Mary Claire has discovered molecular markers. We have immunophenotyping is there. Immunophenotyping, flow cytometric analysis, and immunohistochemistry, histochem. These I will discuss. So there are multiple methods of detecting cancers. Okay. So we have seen few of the methods. The next one, we have discussed about classification of cancers. Classification of cancers. How the cancers has been classified, tell me students?
Based on progression.
Based on tumors, lymph nodes and metastasis. Based on T and M classification. Based on number of tumors, number of nodes and the rate of metastasis we have classified. And we have how many stages of cancers?
Four.
Stage 1, stage 2, stage 3 and stage 4. Write down properly just this classification. Stage 1, okay, there is a 1 or 2 tumors, no nodes and no metastasis. This is stage 1. Only when you are writing you will remember, that's right. Stage 1 is clear, okay. There are some microtumors. Only microtumors we detected means that is stage 1. There is no nodes involvement and metastasis involvement. Then coming to the stage 2. In stage 2 what happens is, so T3 tumors are almost 3 tumors, okay? More than 2, okay? 3 tumors, no nodes and no metastasis. Still this is stage 2, okay? So stage 2 are 2 types, stage 2A and stage 2B. Okay? If the tumors are more than 4, then that is stage B. Okay? More than 2 tumors, stage 2A.
Notes 0 नहीं होंगे sir. Zero.
Sorry, tumors 4. Nodes are 0. Okay? But the number of tumors are increased. In stage 2, number of tumors are more than 2. More than 2 tumors is stage 2. Okay, but still there is no node and metastatic involvement. Okay, then third one, there are tumors, 1 to 2 tumors, 1 node involvement and no metastatic. This is A, stage A. Stage, okay, stage 3A. Stage 3A, 1 to 2 tumors, 1 node involvement is there. When 1 node involvement is there, that is A. Then stage 3B is tumors 3 to 4 plus a lymph node more than 1, that means 1 to 2 and no metastatic. Still there is no metastasis in stage 3. Then C, tumor 4 to 5, then nodes more than 2, okay, 2 to 3, then metastasis 0. That means stage 3, the node involvement has been started. The node spread has been started in stage 3. So when we say stage 3 cancer, that means cancer slowly started spreading. But it didn't spread throughout the body. Cancer spreading in the lymph nodes but not spreading into the circulation. Is it clear?
Yes.
Yeah. Then last and final, very sad, that is stage 4. Here there will be metastatic involvement. Okay? Maybe tumors will be 3 to 4 plus lymph node will be 1 to 3 plus metastatic 1, metastatic 2, metastatic 3. Okay. On metastasis will start exhibiting or will show in stage 4 cancers. Okay, their aggressiveness will be seen in further classification ABCD. Is it clear? So these are the things. Until here, until the cancer in this stage is called as benign. Once the cancer become a metastatic, this will become a malignant, very dangerous. Okay, a malignant cancer, treating a malignant cancer is very tough. Okay, so that's it. Is this clear? So,
Sir?
so far we discussed these things.
Sir?
Can we also
Sir?
emphasize on
Stage 4?
stage 4, apart from tumors and nodes, there will be a metastatic involvement. Metastasis 1, 2, 3. The severity of spreading of cancer. Metastasis means spread of cancer from site of origin. Cancer is in ovary, but now I can see ovarian cancer cells in brain or in the lungs. That indicates the metastasis. It's migrated. Okay? Yeah. These basics you understood. Then next basic I said is a cell, a cancerous cell. Keep in mind, gene mutation lead to some altered protein, and that altered protein will be expressed on the cell surface. Right? These protein markers. Yes. These protein markers, when we are detecting protein markers in a cancer, these protein markers are called as phenotypes. Protein markers are called as phenotypes, whereas the genetic markers are called as genotypes. Okay, keep in mind, I will give one example. A normal helper T cell will have a normal gene. That normal gene will create a normal protein called as cluster differentiation 4. CD4 is a normal protein in helper T cell. Once the helper T cell got a cancer, okay, a lymphoid cancer, lymphoid leukemia, during a leukemic condition, the same helper T cell will produce some other cancerous markers, 28 or something. Okay, when we detect CD24 or 28, that indicates it became a cancer. When we detect CD4, that indicates it's normal. So now you understood, we can detect cancers either at the level of phenotypic expressions or at the level of genotypic expressions. Okay, we will cross verify both. We will see PCR studies, that is genotype, and flow cytometric studies in phenotypic expressions. Flow cytometry is a device that is used, okay, that blood counter. Similarly, rather than blood cells, we can detect any cells, that is flow cytometric technique. Anyway, up to here is clear, right?
No.
Yes.
No. Yeah, where are you confused?
There. The last parts I didn't get it.
Okay. When by seeing the cell surface, if I can identify, that is phenotype. Okay. If I am unable to identify by seeing the cell, rather than I need to study the cell genetics, then that will be genotype. Okay. Sometimes a cancer cell will look like a normal cell. Sometimes a cancer cell looks like a normal cell. Then how to identify? We need to take the genetics. We need to see the gene sequencing. That's it. Okay. Very simple. Nowadays, gene sequencing became very cost effective. Previously, complete gene sequencing is around 1 lakh. Okay. If I want to see complete sequence of Abhiram, I need to invest to 1 lakh. But recently, I did my son's sequencing, that is around 22,000. 1 lakh to 22,000 means it's a huge reduction and there is a huge advancements in the genetics, okay, outside. Yeah, so genome sequencing, once we do the genome sequencing, all the diseases can be seen in the body. Even after 40 years, whether Abhinav will get diabetes or not, I can see by genome sequencing. After 40 years, that is the significance of genes. Because he was genotypically, he is pre-exposed to diabetes. Currently, he might not, okay, not he, normally. Okay, clear, right?
Yes.
Yeah. Then next one, the types of diagnosis, I already said, okay. We will see the blood cells, okay, peripheral cells, whole blood, biopsies. Then we are seeing molecular expression, that means the genes we are seeing. We are seeing immunophenotyping. Immunophenotyping means very simple. This is a cell has an antigen, that antigen will be detected by a antibody, okay? If this antibody has any color, that will, if it has a color, that will become ELISA technique. If it has any radioisotope, then that will become radioimmunoassay. When we are detecting the colored tag of this antibody, okay, by some lasers, then that will become flow cytometry. So there are multiple methods of detection of cancer markers in the body. Okay. Then rather than cell, when you are taking a tissue to see the same antigens, say for example in case of Mary Clay, we have seen BRCA. BRCA is actually genetic marker. Okay. When we are using a tissue, to identify a marker, that technique is called as immunohistochemistry. In immunohistochemistry, we are using antibodies on the tissues to see some chemical reactions. Okay, they will give colored products. Okay, this is immunohistochemistry. Then, these are the various methods of detection of cancer markers. Okay. Then genetic, I said PCR. Okay. Genetic markers can be seen by PCR, polymerase chain reaction. There is this advanced polymerase chain reaction called as FISH. Very interesting. FISH. Fluorescein in situ hybridization. Fluorescein in situ hybridization. Very, very important. In fluorescein in situ hybridization, the cell will be alive. We will keep the cell alive and we can see the chromosomal movements. Okay. That is called as in situ. When the cell is alive, we are doing some hybridization. Hybridization means I am keeping some dyes inside a fluorescein dyes inside the gene markers. Okay. Very advanced technique. The inventor of FISH got Nobel Prize. Because of FISH, we understood cancers very well. Because of this FISH technique, we understood cancers. Okay. Because of PCR, we understood the sequencing of whole genome sequencing. There are good scientists actually. They got Nobel, he got Nobel. And another interesting thing. Okay, the number of chromosomes can also cause cancers. Do you remember non-disjunction yesterday? The number of chromosomes, 46 chromosomes or 45 chromosomes. Okay, usually in an adult we have 46 chromosomes, but there are conditions where the chromosome number will be reduced. Okay, the counting of chromosomes is called as karyotyping. Write down the word. The counting of chromosomes. The counting of chromosomes is called as karyotyping. You know, previously when Mendel discovering these genetics, they said karyosomes. Karyosomes means nucleus only in other Greek or Latin. Then since we are seeing the nuclear parts, that is karyosome or karyotyping. Okay. Nucleus is also called as karyosome. So then karyotyping. Alright. Is everything clear? Shall I move forward now?
Yes.
Yeah. The next topic will be the difference between AML and CML. So difference between AML and CML. So I will write here. Difference between acute myelolymphoproliferative leukemia and chronic myelolymphoproliferative leukemia.
Okay, what's up?
Yesterday we already discussed, right?
Yes.
But still, I know you. Yeah. See? They are fed up with repetitions. Anyway, rapid, chronic.
Yes.
Immature cells, mature cells. There are
Genetic markers.
genetic markers. What are the genetic markers we have?
FLK3, NPM1,
And here?
NPM1.
Yeah.
Philadelphia.
Then signs and symptoms. Do I discuss signs and symptoms?
Yes.
What are those?
Fatigue, fever, infection.
Yeah, fatigue, liver. Here, night sweats.
Wait.
Chronic night sweats can be seen. Then chemotherapy is ideal treatment. Here,
Drugs,
we can do
that's
inhibitors. What inhibitors we can do? Tyrosine kinase inhibitors can be given as a therapy for CML. Very good. Then, listen. Prognosis of treatment. Prognosis means the response, okay? The healing response of the patient during the treatment. The prognosis is very good in CML, but prognosis in AML is very less, okay? Even though we are treating the patient, the chances of survival of this patient is very less compared to these patients. Okay? Yeah. This is the difference between AML and CML. The next one, write down types of diseases, types of AML and CML. There is a collective term called as myeloproliferative disorders. Types of myeloproliferative disorders. types of myeloproliferative disorders. Types of myeloproliferative disorders. Okay. Myeloproliferative disorders are also called as myeloproliferative neoplasms. Same word, okay? Myeloproliferative neoplasms. Both are same. Keep in mind when we came up to the level of proliferative, okay, when we are saying something myeloproliferative, that is cancer. We can't say anything. We can't do anything. Anyway, the first disease, write down. Yeah. Now, how we are diagnosing, how we are detecting this means by three parameters. Write down the three parameters. Based on three parameters, we are detecting these three. The three are one, cellular lineage. Cellular lineage. From what lineage those cells has been derived? Myelo, lympho. Okay. Then clinical features. We need to cross-verify clinical features. Then third one is specific gene mutations. Specific gene mutations. With these three criteria, we are going to differentiate various types of myeloproliferative disorders. Now, in your notebook, write down like this. Okay, keep right side space and write down all the disorders. There are almost 7 disorders. Okay. You leave a space here. Write disorder name here and leave space. That we'll see later. Okay. Write down the disorders.
Sir,
the first one.
What is the third?
Third one, specific gene disorders. Mutations. Specific gene mutations. Cellular lineage. Cellular lineage, clinical features, and specific gene mutations. Write down the first disorder. The first disorder is chronic myelogenous leukemia. Chronic myelogenous leukemia. Chronic myelogenous leukemia. CML. Then second one, polycythemia vera. Polycythemia vera. What are these? I will discuss later. Okay. First, write down the diseases. Polycythemia vera. Then third one, SNVER vera. Okay. That is this is also called as rubra vera. That I will discuss later. Okay. Polyrubra vera. There is a condition. Then third one, essential thrombocytopenia. Thrombocytopenia. Essential thrombocytopenia. Enough. Essential thrombocytopenia. Essential ET. This is also called ET. Okay. Essential thrombocytopenia. Then fourth one. This is myelofibrosis. Myelofibrosis. Fourth one is myelofibrosis. Then fifth one. Chronic Fifth one is chronic neutropenic leukemia. Chronic neutropenic leukemia. Chronic neutropenic leukemia. I made a mistake. Neutrophilic. Not clinic, it's a philia. Okay, cancer increased. Neutrophilia increased, penia means decreased. Okay, next one. Sixth one. Chronic eosinophilic leukemia. Chronic eosinophilic leukemia. Chronic eosinophilic leukemia. Then last and final, seventh one, systemic mastocytosis. Systemic mastocytosis. Is it clear? Now we will address each condition. Okay. Now what we are going to do is very simple breakdown. Pay attention. For every disease, when you are writing answer in the exam, also keep in mind, I need only 3 things for every disease. I need the disease definition. What is that disease? What are the second one? Symptoms. Then diagnosis. In diagnosis, what you need to write is what you will observe in a peripheral blood smear. Then what gene marker? Okay, I need phenotypic marker. What phenotypic marker you will detect and what genotypic marker you will detect. what phenotypic marker you are going to detect in that disease, what genotypic marker you will detect, then is there any treatment? If there is treatment, I need the treatment simple statement. Okay, any other lab test, any other lab test if you have. We are not going to study in detail oncology, that is study of cancer. We are only seeing the lab diagnosis aspects of cancers, okay, and hematology point of view. Okay, the first one, what is the first disease? Chronic myeloid leukemia. So, as the name states, it's a chronic condition affecting the myeloid lineage leukemia. Okay. Now listen. In chronic myeloid leukemia, there is a we need to do important test hyphen right now. In chronic myeloid leukemia, hyphen detection of Philadelphia chromosome. Detection of Philadelphia chromosome. The presence of Philadelphia chromosome is the characteristic feature for chronic myeloid leukemia. Philadelphia chromosome. Okay? Philadelphia chromosome has a technical term. The marker has a technical term. That marker is called as BCR-ABL1 marker. BCR-ABL marker. This marker is called as BCL-ABL marker. Okay. I will tell one interesting thing. When I did my son's genome sequencing, the only thing doctors focusing is they have some 2000 sets of problematic primers or markers. They just run all the markers in the blood sample, whether any marker detected or not. Luckily, there is no markers in his body. That means he is fit. Okay, that is the significance of PCRs. We know the markers. How we get to know the markers? Because of scientists like our Mary Claire. They discovered the markers for particular conditions, and we have standardized the markers. Okay. So chronic myeloid leukemia can be detected by a marker called as Philadelphia chromosome. That Philadelphia chromosome has a marker called BCR-ABL. Okay, that's it. Yeah. Then next one, polycythemia vera. Okay, what is polycythemia vera means? It's an abnormally increased red blood cell count. Okay, there is an abnormal increase in red blood cell count, more than 7 million per cubic millimeter of blood. More than 7 million cells per cubic millimeter of blood. Okay, so overproduction of red blood cells. That is also a cancer condition. Okay, polycythemia vera. So there are some things. And write down the marker. The marker for polycythemia vera. JAK2, JAK, JAK2 marker, JAK2 kinase marker. JAK2 kinase marker. Detection of JAK2 kinase marker, and V617F marker. V617F marker can be seen. Okay. V617. There are some technical terms. Okay, that's it. And that's it. They themselves have further guys. At least you try to remember in polycythemia vera there is an increased RBC count and they are the markers. Okay. So patient exhibit splenomegaly, then abdominal pain, then there will be rashes on the skin, technical, hemolytic features. Yeah. Polycythemia rubra. So the advanced stage. Okay. A severe form of polycythemia vera is polycythemia rubra, where the patient will exhibit swelling of hands with red spots. Rubra means rubor. Rubor means red spot with swelling is called as rubra. Okay, that's it. So that's it. This is all about polycythemia vera. Then third condition. Third condition is essential thrombocytopenia. Thrombosthenia. Okay, essential thrombosthenia. That is the third condition. Yeah, thrombosthenia. Okay, sthenia or penia same. Okay, thrombosthenia. So what happens in essential thrombosthenia? There will be an abnormally increased platelet count, but all those platelets are worst. That means they are dysfunctional, not good platelets. Okay. Yeah, write down the markers in this one. JAK2, CALR, MPL gene markers. Again, JAK2, then a CALR marker and MPL gene marker. This is for thrombocytes, okay? Thrombocytes. Thrombocyte markers are CALR, MPL gene marker, JAK2, CALR and MPL gene markers. And also, we can see increased clotting time, increased bleeding time, headaches in the patient, and also vision abnormalities can be seen. I was surprised to know this fact. During this condition, the blood will start oozing in the eyes. So you just imagine, rather than clear tears, you will see red blood in the eyes. So the world will be red.
Is it a one mark
Will
or
be red
something?
filter. Because there is a bleeding in the eyes. Very painful.
Sir, that's CLL, MDL, that's a one mark or something?
CALR is one comma, then MPLG. There are three markers. JAK marker, CALR marker, and MPL marker.
GI marker is different in
Yes, they are different. Next one. I know this class looks boring, that's why I was trying to keep it entertaining.
It's not boring.
Yeah, the next disorder is, what is the next disorder? Myelofibrosis. So listen students, there is a cells called as fibroblasts. Fibroblasts will heal the scar tissue. Okay? When you get a cut, that healing has been done by fibroblast. It's a type of skin cell. Now in this condition, the bone marrow will be filled with fibroblast cells. Okay? There will be a bone in bone marrow. There is a bone in the bone marrow. You got my point, right? So this is the bone marrow, right? We know this is the bone marrow. In this bone marrow, there will be another bone. And inside that bone, there will be marrow. So bone in bone marrow, okay? Yeah, you can understand. You just imagine, you got an egg, you crack it, but there is another egg inside an egg, okay? That is this condition. So the bone has been calcified, that is the bone in bone marrow condition. Yeah, myelofibrosis. Again, myelofibrosis also has same markers like essential thrombocythemia. Okay, JAK2, CALR, MPL gene mutations. JAK2, CALR, and MPL gene mutations. All has been seen. Okay, and also the red blood cells in myelofibrosis. In this condition, the red blood cells won't be smooth, they will be like this. The red blood cells looks very rough. Okay. In myelodysplastic myelofibrosis, the red blood cell looks rough. In PBS, in peripheral blood smear, the red blood cell looks rough. Okay. It will be porous, granulous, porous red blood cell will be seen in myelodysplastic syndromes or myelofibrosis condition. Okay. Yeah, by the way students, if you think these tests are less cost, they are not less cost. Myelofibrotic MPL test, okay? CLR MPL test is around 1,000. So to detect these cancer markers, the tests are very costly. Okay? Yeah, they are very costly actually. Yeah. Fine. Next one. What is the next condition? Chronic eosinophilic leukemia, right? Chronic eosinophilic leukemia. So this is chronic eosinophilic leukemia. Write down the markers. The markers for chronic eosinophilic leukemia. These are the markers. PCM1 JAK2, ETV6 JAK2, and PCM JAK2. These are the three markers. And there is one more marker, TET2. Occasional cases JAK2 mutation, but AXL and TET2 are both common. So either we can see those markers or we will see these markers. Okay. So write down at least three markers for this condition. Essential eosinophilic leukemia markers. Write down those three markers and this and this marker. So the peripheral blood smear expression, you will see small cells, okay, scattered spots of white blood cells. This is the eosinophilic leukemia condition. Next one. Last and final, mastocytosis. Okay, I already given this one right to you. Write down this disorder also. This is mastocytosis.
Neutrophilic also.
Yeah, neutrophilic, right? Neutrophilic markers write down. Neutrophilic markers again JAK2 mutations. Even in neutrophilia, JAK2 mutations will be observed. JAK2 mutations can be observed in neutrophilia. Okay. Listen now. Have you seen a trend in the markers? Almost all myeloid series have JAK2 mutations. So just having a JAK2 mutation can give a myeloid leukemia, but we need to pinpoint it. Is it a polycythemia? Is it a thrombocythemia? Again, there is a confusion. See. Okay, last and final, mastocytosis. So this is mastocytosis. There is an abnormally increased abnormal granulocytes in the body. Abnormally increased abnormal granulocytes in the body. Okay. So the patient actually, his color is that palm color, but he became very reddish. Okay. This is a cancerous condition. So yeah, these are the various types of myeloproliferative and myelodysplastic syndromes. Okay. There will be an abnormal cells, and these are the various markers for various conditions. Sporadic, there are age-associated MDS and non-age-associated MDS, and then markers.
Last one is marker.
Okay, for mastocytosis there are multiple markers. That's why I excluded it actually. Okay. Can you able to understand one thing? Cancer doctors are like scientists only. They are doing research, and from patient to patient, the markers are different. So pinpointing the exact cause of the type of cancer in patient is very tough. Okay, because all cancers have random mutations, and random itself you can understand. Each patient has, okay, just like your fingerprints. Each patient. Okay, cancers are unique to patient. Okay, so it is very challenging to treat these cancers. And one more problem, we are literally targeting our own genes to treat these patients. That means even healthy cells will get affected because of treatment, chemotherapy. Okay, so chemotherapy means we are partially killing the patient, but maintaining patient is alive. Okay, very tough.
Sir,
yes.
what are these markers?
What are these markers? See, mutations. I said right. Anything that we will detect to connect it with the disease is a marker. The marker for fever is body temperature. Got my point? The definition of marker is something that we can identify or connect it with the disease. Here, when we see, I have taken blood sample, I have extracted his DNA, and I started studying the DNA pattern, okay, the sequences of DNA pattern. There I know that if the patient has one particular marker, A, T, 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 G, G, C, there is a marker, this marker is BRCA marker. BRCA marker. So if I detect this sequence in the patient, that indicates this is a marker of BRCA, that indicates he or she might have a breast cancer. So similarly, we have various markers for various conditions. Simple. Okay, not these markers. Okay. Yeah, thank you very much. This is all about myeloproliferative. Next time we'll see lymphoproliferative. Thank you. That's it.
That's a confusing lecture. What the hell? That is so confusing.
Markers. No need to write these markers, okay? Leave it. Tomorrow I will discuss another interesting topic. Maybe on Friday.